Welcome to the C2 Pipeline Innovation and Curiosity Center Sheep Heart Dissection Lab. By the end of this lab, you will be able to demonstrate proper dissection safety techniques, identify the right and left sides of the heart, identify the different structures of the heart, measure the walls of the left and right atria and ventricles, and compare and contrast the structure of a sheep heart to a human heart. Safety glasses, gloves, and closed-toe shoes are a must for any dissection lab. You should also have your hair pulled back and your sleeves rolled up. If available, you should wear a lab coat. The tools that you will be using to dissect are very sharp. Great care should be taken when using the tools. Never walk around the lab with a tool in your hand. When you're handling the heart, be gentle with it so that you don't accidentally squirt any fluids on your face or body. Lastly, this movie is set up to give you the necessary directions for the lab. Please do not perform any actions while the movie is playing. You should be watching and listening to the movie. When the facilitator pauses the movie, he or she will give you additional directions as necessary. Studying the sheep heart allows us the opportunity to examine a heart that is structured just like our own. Sheep have a 4-chambered heart. Investigating how blood pumps through the sheep heart will allow us to examine how our hearts keep us alive. Most heart diagrams show the left atrium and ventricle on the right side of the diagram. Imagine the heart in the body of a person facing you. The left side of their heart is on their left, but since you are facing them, it is on your right. Remember to watch and pay attention to the video while it is playing. Do not perform any actions until the video is paused. Identify the right side of the heart from the left by using 2 references: the diagonal line of blood vessels that run on the surface of the heart and the apex. The diagonal line provides a reference point that can be used to divide the heart into 2 sides. Additionally, the apex or pointed end of the heart will aid in identifying the halves as well. The half of the heart that includes all of the apex is the left side. One way to confirm that you have correctly identified the right and left side is to squeeze each half. The left half will feel much firmer and more muscular than the right. The left side of the heart is firmer and more muscular and therefore stronger than the right because it has to pump blood to the whole body, while the right side only pumps blood to the lungs. Before you squeeze the heart, make sure that it is facing down so that any fluids do not squirt out at you. We will pause the video at this time to identify the sides of the heart. When the video is paused, you'll turn your heart so that the right side is on your right, just as if it were in your body. You'll examine the flaps of darker tissue on the top of the heart. These ear-like flaps are called auricles. The auricles of the heart are wrinkled portions of each atrium that protrude externally to form a pouch. This structure is sometimes referred to as the atrial appendage. The auricles function as a reservoir for the atria. The term atria is plural for atrium and refers to both the right and left atrium collectively. When the video is paused, you'll turn your heart over and locate the left and right auricles. We will pause the video now. Before we go on to identifying the major vessels of the heart, let's compare and contrast the 3 types of blood vessels. A blood vessel is a hollow tube that carries blood in animals, including humans, around the body's systems, allowing for proper oxygenation of tissues including muscles and nerves. Arteries, veins, and capillaries are all different kinds of blood vessels. Arteries are blood vessels that carry blood away from the heart. The aorta, right and left pulmonaries are examples. Arteries are the higher pressure part of the circulatory system, as they are getting blood from the heart and sending it out to other parts of the body. Arteries are fairly large, elastic, and muscular. These vessels are 3 layers thick, as they have to withstand the high pressure of blood being pumped off the heart. These are the strongest of the 3 types of vessels. Veins are blood vessels that carry blood back to the heart from the body. Veins carry deoxygenated blood back to the heart, although this is not the case with the pulmonary vein, which carries oxygenated blood to the lungs. Veins are tubes that collapse when not filled with blood. Like arteries, veins have 3 layers, but they're not as thick or tough since they do not transfer blood at such a high pressure as the arteries. Both veins and arteries are most easily defined by the direction of blood flow rather than by oxygen content, although oxygen supply is generally high for arteries and low for veins. Remember, arteries move blood away from the heart, while veins move blood toward the heart. Capillaries function to facilitate the exchange of various things between blood and tissue. They act as a middleman, receiving blood from the arteries rich in oxygen, exchanging it for carbon dioxide from surrounding tissue, and then dumping the waste-rich blood out to the veins in order to get it back to the heart for cleansing and recirculation. Capillaries are very thin and fragile. What's interesting is that veins run parallel to the arteries throughout the body, with capillaries forming a web-like network between them. When the video is paused, you'll place your heart in its original position with the left atrium and ventricle on your right side. You'll locate the large opening at the top of the heart next to the right auricle. This opening is the superior vena cava. The superior vena cava brings blood from the top half of the body to the right atrium. When the video is paused, you'll stick your finger down this vessel and you should feel it open into the right atrium. We will pause to locate the superior vena cava now. The inferior vena cava brings blood to the heart from the lower part of the body. It is a little down and to the left of the superior vena cava. When the video is paused, you'll insert your thumb into this vessel and your index finger into the superior vena cava. You'll know that you have inserted your thumb into the inferior vena cava as your thumb and finger will be able to touch. Your thumb should lead into the right atrium along with your index finger. We'll pause at this time to locate the inferior vena cava. While observing the inferior vena cava, you may have also noticed another blood vessel next to the left auricle. This is the pulmonary vein. The pulmonary vein brings blood from the lungs into the left atrium. Remember, the pulmonary vein is located right next to the left auricle. We'll pause to locate the pulmonary vein at this time. The aorta takes oxygenated blood from the left ventricle to the rest of the body. There is a right and left ventricle referred to collectively as ventricles. The ventricles are the lower chambers of the heart. The aorta branches into more than one artery right after it leaves the heart, so it may have more than one opening in your specimen. When the video is paused, look carefully at the openings and you should be able to see that they are connected to each other. We'll pause to locate the aorta at this time. Just behind and to the left of the aorta, there is another large vessel. This vessel is called the pulmonary artery. The pulmonary artery takes blood from the right ventricle to the lungs. Notice in the picture that the left atrium is on the left side. This means that the side of the heart that is facing you is the dorsal or back side. We will pause to locate the pulmonary artery. Woohoo! Time to cut. Now that we have observed the heart externally, let's begin to look at its internal structure. When the video is paused, you'll insert your dissecting scissors or scalpel into the superior vena cava and make an incision down and through the wall of the right atrium and ventricle as shown by the dotted line in this picture. As you're doing your cutting, you will be cutting through the whole wall so that you are able to spread the two sides apart when you are finished making this cut. We will pause to make the cut at this time. Now that you have made the incision, we'll look at the inside of the right side of the heart. Remember not to perform any actions until the video is paused. When the two sides are pulled apart, you'll look for 3 flaps of membrane. These membranes form the tricuspid valve between the right atrium and the right ventricle. The tricuspid valve was named so because it has 3 cusps or flaps. This valve allows blood to enter the ventricle from the atrium, but prevents backflow of blood from the ventricle into the atrium. Chordae tendineae, or heartstrings, are the tendons that connect the papillary muscles to the membranes of the valves. The valve acts much like a door that opens in only one direction. The tricuspid valve is one of 2 atrioventricular valves, also called AV. The right AV is called the tricuspid valve. At this time, we'll pause to locate the listed items. Remember that the pulmonary artery is just behind and to the left of the aorta. After the video is paused, you will insert a probe into the pulmonary artery and it should come through to the right ventricle. You'll know you're in the proper vessel if it comes through to the right ventricle. We will pause to place a probe into the pulmonary artery through to the right ventricle. The second incision will open the left atrium and ventricle. When the video is paused, you will make an incision through the pulmonary vein to open the left atrium and ventricle. As with the right side, you will cut through the pulmonary vein and the wall of the heart so that you are able to open the left side of the heart. We'll pause at this time to make the incision. When the video is paused, you'll locate the mitral valve or bicuspid valve between the left atrium and ventricle. This will have 2 flaps of membrane connected to papillary muscles by the chordae tendineae, just like the tricuspid valve. The mitral valve is the other atrioventricular valve that prevents blood from flowing back into the atrium from the ventricle. You'll also locate the chordae tendineae, papillary muscle, and the left ventricle. We'll pause at this time to locate the listed items. When the video is paused, you'll make the final incision through the aorta. First, you'll insert a probe into the aorta and observe where it connects to the left ventricle. Then you will make an incision down through the aorta and examine the inside carefully for 3 small membranous pockets. These form the aortic semilunar valve. This prevents blood from flowing back into the left ventricle. When you make this incision, you will be opening the aorta. We'll pause at this time to make the incision. After the video is paused, you'll locate the semilunar valve and the cusp of the valve. You'll examine the inside carefully for 3 small membranous pockets. These form the aortic semilunar valve, which prevents blood from flowing back into the left ventricle. We'll pause at this time to locate these structures. When the video is paused, we'll do some measuring. You'll use your ruler to measure in centimeters the left and right atria and the outer wall thickness of the left and right ventricle. Remember, you'll be using centimeters. We'll pause at this time to measure these parts. This concludes the sheep heart dissection. We hope you have enjoyed your lab and have a better understanding of how your heart works and how the sheep's heart works.